What is this virus that's causing a worldwide pandemic, the COVID-19? How to understand something that we never seen before? The first action is to understand the genome. Genome is the genetic material of a species. It's a nucleotide sequence that I will talk about later. By studying it, we found that this virus belongs to the coronaviridie family, known for causing common disease as the cold. Also, that its origin was in China and is very close to the genome of the coronavirus bat SLCoVCC45 of the bat species Rhinolophus cilium. But how a virus that has never been present in humans is causing so many problems? How was discovered that a single virus managed to travel all of the world and I didn't? We also know the mutations of the virus present in bats created a protein called spike that facilitates the binding of the virus to a receptor in our DNA, receptor that produces an enzyme responsible for regulating blood pressure and vasodilation in the lungs. We know all this information by discovering the COVID genome. But how is that? First, a DNA or RNA is formed by nitrogenous bases. Its sequence is responsible for the production of all proteins present in organisms that forms the whole body. In sequencing, we identify the sequence letter by letter. For sequence, a technology called Genome is used. In Brazil, this discovery was made by the saliva sample of a São Paulo's resident, the first infected in the country. In virus, the genetic material is RNA, and the sample that containing it is applied in the genome with the enzyme transcriptase reverse. After, the enzyme binds into the RNA, producing the reading. And then, with this reading, it produces the sequencing of the referring DNA. But why translate the RNA into a DNA sequence? RNA is composed of just one strand. It's essential for the sequence and production of proteins and enzymes in living beings. It occurs by RNA binding to DNA in the cell's nucleus and producing messenger, ribosomal, and transporter RNA that in the ribosomes stimulates the protein production. Virus has only RNA and, when in contact with the human body, binds with the cell. In COVID's case, by the spike. After binding, it can cause the altered production of certain proteins. When we turn the sequence of RNA into DNA, we have its completed genome. This complementary DNA is then expanded using the polymerase chain reaction technique (RCP), multiplying it so possible errors are found and excluded, enabling a clearer reading. Understand COVID genome will help us to develop vaccines and drugs for treatment. Thanks for watching and be safe!